Mnemonic, General Motors makes classic cars. GM equals EGG and EGM. Activate the classic pathway. Question 1. Which of the following immune molecules initiates the classical pathway of complement activation? A. Mannose binding lectin. B. IgG. C. C3b. D. Properdin. E. Toll-like receptors. Correct answer, B. Explanation. The classical complement pathway is triggered by IgG or IgM binding to antigen. Then C1 complex binds to the Fc region of the antibody. Mnemonic. General Motors makes classic cars. T equals IgG and IgM, classic. Why the others are incorrect? A. MBL equals lectin pathway. C. C3b equal amplification step, not initiation. D. Properdin stabilizes the alternative pathway, doesn't initiate it. TLRs are part of innate immunity, not the complement cascade. Question 2. Which of the following best describes the initial trigger for the lectin pathway of complement activation? A. Antigen-antibody complex. B. Bacterial lipopolysaccharide. C. Mannose residues on pathogen surfaces. D. IgE bound to basophils. E. C5a binding neutrophil receptors. Correct answer C. Mannose residues on pathogen surfaces. Explanation. The lectin pathway is activated when mannose binding lectin, MBL, binds to mannose or sugars on the surface of microbes. Why the others are incorrect? A. Antigen-antibody, classical pathway. B. LPS, alternative pathway. D. GE, allergic responses, not complement. E. C5a equals chemotactic factor, not an initiator. Question 3. A researcher isolates a bacterial strain that triggers complement activation without antibodies or sugar recognition. Which pathway is most likely being activated? A. Classical pathway. B. Lectin pathway. C. Toll-like receptor cascade. D. Alternative pathway. E. CD40-CD40L signaling. Alternative pathway. Explanation. The alternative pathway is activated directly by microbial surfaces, including bacterial products like LPS, without the need for antibodies or mannose recognition. Why the others are incorrect? A. Classical requires IgG-IgM. B. Lectin requires mannose or sugar residues. CTLRs equals pattern recognition, not complement pathway. E. CD40-CD40L involved in B-cell activation and isotype switching, not complement. Let me know